G'day everyone, thanks very much for joining me at 10 Drugs That Changed the World. I'm Phil Burcham, and although I look really youthful and dynamic, I've actually got 30 years experience teaching pharmacology. Pharmacology, of course, is the branch of science that studies how medicines work, how they produce their healing or therapeutic effects on the body. Pharmacology is a big part of the education of doctors and pharmacists and dentists and other groups of health professionals. And the point of that pharmacology education is to help prescribers and other users of medicines use them wisely. That is, how do we maximise the benefits that modern pharmaceuticals can bring to suffering patients, but at the same time minimise the harm that they might cause to individuals? The aim is to avoid underdosing and overdosing. Now, while pharmacology is a great subject to teach, one of the limitations to me personally is because I'm the sort of person that likes to know a bit about where new medicines come from, what are the historical origins of new breakthroughs in medicine. We never really get to talk about those topics much in delivering a pharmacology course to doctors or future dentists or pharmacists, because we're really focused on understanding the scientific aspects surrounding drugs as well as the clinical ramifications of using them in patients. So one unanswered question that we never get much time to address is, where did the medicines in current use come from? What we want to do in this block of videos is break some new ground by exploring some of the fascinating historical, scientific and human dimensions of the pharmaceutical innovation sector. I think this is really some fascinating content that we're going to be covering. I find it very exciting and interesting, and I hope you will also if you hang around to follow this video series through to the end. You'll find that it can be pretty inspiring to go back and look at the origins of some of the major history-shaping pharmaceuticals that were introduced in the 20th century. But one of the reasons I'm doing this and going to this trouble is one of the things I've noticed as a person who's now in the wrong end of his 50s is that when I was a boy in the 60s, pharmaceutical companies used to enjoy very high public standing. In fact, some of the highest rates of public approval were actually for drug companies within the pharmaceutical sector. That's not necessarily the case now, and many people wonder if a lot of pharmaceutical innovation is driven by commercial concerns, wanting to make more money and deliver value to shareholders. All those things are very important, of course, to making sure that the enterprise remains healthy and financially viable for the next generation. But at the same time, I think it's really good to go back to look at the origins of some of the great discoveries in this field of science, because we'll find that people were usually motivated by, first and foremost, a concern for the well-being of suffering human patients. And I think that going back to explore one's roots is always good for the soul, and hopefully this little video series will be of value to you as you think through your place in a modern world where pharmaceuticals are a big part of daily life. Okay, so what we want to do then in this historically attuned block of videos is to explore 10 world-shaping medicines, from morphine to chloroform to penicillin, and even some of the bad guys of the pharmaceutical innovation historical process, such as thalidomide. And hopefully we'll see how the discovery of some of these significant pharmaceuticals had a profound, world-changing impact that we still benefit from in very real ways long after these discoveries. And also we're going to see how some of these discoveries accelerated and gave momentum and inspiration and coherence to the whole process of medical innovation. But before we can start looking at some of that fascinating historical content, I'm going to take some time in the first half a dozen or so videos in this series to unpack some basic scientific concepts that we use in pharmacology to understand the effects of drugs on the human body. I think it's important that we do this because we could jump into the historical stuff and the inspirational human discovery stuff first, and that's all very interesting, but I think to really appreciate some of the struggles and the achievements of some of those great figures in pharmaceutical innovation history, we really need to take a bit of time to think through some of the scientific challenges surrounding the effective use of medicines in humans. So what I want to do in the first half a dozen or so videos is look at some of these very basic concepts in modern pharmacology. And hopefully these will be beneficial to you, because if you use pharmaceuticals personally, or perhaps your grandma or your mum or dad are on some sort of pharmaceutical, understanding a little bit of the science surrounding the use of those products can be good for all of us in helping to make sure that we use these remarkable products, but potentially harmful products, very safely as possible. Okay, so what I want to do in this first episode, in what remains of this video, is to look at some basic terminology that we use in modern pharmacology. And there's two terms I want to get across to you that I think are really important, and once you understand the rationale for using these terms, we'll be well on our way to making progress in understanding modern pharmacology and how we approach thinking about drugs scientifically. The first term I want to convey a basic appreciation for is the notion of a therapeutic window, and then also the second concept is the term pharmacokinetics, which tries to explain what happens to drugs while they're in the human body. So there's a couple of unfamiliar terms there probably for you, but hang on, don't worry, I'll unpack them as the rest of this video develops. Now, one of the ways of seeing the relevance of what I'm going to talk about in this particular video is just to think about your common exposure to pharmaceuticals on a daily basis. You might know people, or perhaps you yourself have used a particular pharmaceutical, and you've noticed that sometimes the prescription requires you to take the pills very frequently, such as some of the antibiotics, such as carbenicillin and the related ampicillin. These sorts of drugs need to be taken either every 6 hours or every 8 hours, so very regularly, quite an imposition on the life of a patient. On the other hand, there are other drugs such as disopyramide used in the treatment of rhythm disorders of the heart, which only is taken in a morning and an evening on 12-hour intervals on a twice daily basis. Then there are many other medications where we only take them once daily at 24-hour intervals. But then there are some medications such as methotrexate, which are only taken once weekly, particularly in the treatment of rheumatoid arthritis. And then there are others such as risedronate, used in the treatment of various skeletal conditions. It's used even on a monthly basis. So what's going on here? Why is it that some drugs you need to take many times every day at regular intervals versus some you only take on a daily, weekly or even monthly basis? Well, the answer to that question of why some drugs need to be taken regularly and some less regularly, or all need to be taken regularly but certainly at different intervals, has a lot to do with their pharmacokinetics. And that's what I want to talk about in this particular video. Okay, so what does that term mean? We've obviously created this term by borrowing terms from other scientific fields, and in this case, pharmacologists are pretty good at this. We've pinched the term kinetics from a chemistry department, and we've tacked it onto the term pharmaco to tell us that we're using that term within the context of drugs and pharmaceuticals. Okay, so what is pharmacokinetics, and how is it related to modern pharmacological knowledge of modern pharmaceuticals? Well, essentially, you can imagine that when we think about drugs, we often think about what they do to the body. So let's say we're taking a drug to alleviate pain or to reduce our blood pressure. Those sorts of outcomes associated with the use of drugs and motivating the use of the medication are really pharmacodynamic. Pharmacodynamics is the branch of pharmacology, a very important domain of pharmacology that wants to know what does the drug do to the body. How does the drug work? How does it produce its effects all the way down from the cellular level all the way through to tissues and organs and whole body systems? How do drugs produce their effects on body systems that are desirable in the treatment of disease? But every drug is a bit like a coin. We flip it over, on the other side we see that there's also another important domain of knowledge concerning any pharmaceutical, and that is its pharmacokinetic properties. And there we're asking, what does the body do to the drug? Okay, so the coin analogy is actually quite helpful, because just if you're a coin collector, and I'm not, but I think I'm familiar with the idea that people who collect coins like to collect coins from different countries and different types of currency, and every individual coin within a currency has its own properties, its own distinctive head and its own distinctive tail. And in the same way in pharmacology, both the pharmacodynamic and pharmacokinetic properties are unique to a particular medicine. And this is why it makes pharmacology such a fascinating field of study, because we have to get our head around so many hundreds, if not thousands, of pharmaceutical agents. Okay, so today, in this particular video, I want to focus on this concept of pharmacokinetics. I want to unpack this idea. What is it and what are we talking about when we just talk about the pharmacokinetic properties of drugs? Okay, so the pharmacokinetics we can define as a branch of pharmacology that studies the behaviour of drugs as they move into the body after dosing a patient, as they move around the body within the bloodstream, getting to particular tissues around the body, and then out of the body in the processes of excretion. Okay, so just as in chemistry, the term kinetics is used to refer to the speed of reactions or the rate of reactions between different molecules. So chemists will put a couple of different chemicals, chemical A and B, in a test tube, and they'll time the reaction between those two molecules, and they'll measure the rate of formation of product C. That's all kinetics from a chemical point of view, but when we use that term in pharmacology, we're really talking about how blood concentrations fall after dosing, or rise after dosing and then begin falling in the case of oral dosing. Okay, so pharmacokinetics concerns itself with describing the scientific properties that determine the speed or rate at which drug levels in blood and tissues rise and fall after dosing with a particular pharmaceutical. Okay, now fundamentally, there are four basic processes, essentially physiological processes, that shape the pharmacokinetic properties of drugs. It's helpful to distinguish each of these, and in fact, I'll come back in future videos and we'll hopefully look at each of these processes in a little bit more detail. Obviously, when you take a couple of pills for a headache, for example, then the active pharmaceutical agent within that pill needs to get out of that pill and make its way into the body. And this is fundamentally the process of absorption. So the readiness with which drugs are absorbed out of the gastrointestinal tract, the stomach and the intestines, is very, very important in achieving effective responses to medications. This is followed by distribution, which refers to the readiness with which drugs are carried by the blood around the body to make sure that they reach effective levels and concentrations in particular tissues of concern. So if we're giving the drug to treat depression or pain, then we want it to achieve certain concentrations in the brain or central nervous system. In the case of heart disease, we want to achieve particular concentrations at the heart tissue. Okay, so they're all fundamentally important questions that we look at when we consider the process of distribution. But then there are two other processes that begin competing for any drugs floating around in the body, because they begin removing the drug from the bloodstream to protect the body against the presence of that foreign chemical. First of these is metabolism in the liver. The liver has a remarkable capability to carry out thousands of chemical reactions on foreign substances. We'll unpack some of those concepts in a future video. It's really interesting. But then there's another process. Sometimes drugs are deliberately or directly pulled out of the bloodstream as they make their way through the kidney. We call this process excretion. And together, these two processes of metabolism in the liver, excretion in the kidney, are both sometimes cooperating. Sometimes one dominates the other, but effectively reducing blood concentrations of that pharmaceutical agent. Okay, so it's because of these two processes that are removing the drug from the bloodstream. That's why we have to give patients follow-up doses to re-achieve effective blood concentrations. But I'm getting ahead of myself. Now, just to orient what I'm talking about today in terms of those four fundamental processes that shape the pharmacokinetic properties of drugs with some other events that are very important when you take a medication, I thought it's helpful to distinguish between what we call the pharmaceutical phase of drug action. So this begins when a patient ingests a couple of pills, and the pills begin a process of disintegration and dissolution, breaking down into smaller and smaller products, which then release those individual drug molecules so that they're floating around in solution within the fluid within your stomach or your upper intestine where most drug absorption occurs. Okay, we call those processes the pharmaceutical phase of drug action. Now, this isn't necessarily of prime concern to pharmacologists because it's not our expertise. This is the sort of thing you'd learn a lot about if you're studying a Master of Pharmacy or a Doctor of Pharmacy, and which includes the study of pharmaceutical chemistry. They spend a lot of time, there's a very specialised topics of making sure that when we manufacture pills, they're properly formulated to release their chemical payload when we want them to within the body and within a particular part of the gastrointestinal tract. But really, the bit we're interested in pharmacology, we might call it the pharmacokinetic phase, begins once those individual drug molecules are in solution in the fluid in the within the stomach or the intestines, and that's when we begin this all-important process of absorption, which then kicks off these subsequent fundamental processes that shape the ADME properties, the absorption, distribution, metabolism and excretion, that overall shape the pharmacokinetic behaviour of a drug. So essentially, coming back to that question I raised a few minutes ago, why is it that drugs such as carbenicillin and ampicillin need to be taken 3 or 4 times daily versus something like risedronate, which is only taken once monthly? The answer really lies in these processes of absorption, distribution, metabolism and excretion, which shape the rise and fall of drug concentrations in blood and tell us how often we need to replenish the drug by giving new doses of the pills and at what time intervals. Okay, so that really brings us to the heart of pharmacokinetics, really, which in terms of being able to say anything intelligible at all about the pharmacokinetics of a drug, we really need to know what we call its blood concentration versus time profile, to know how concentrations of the drug rise and then fall after dosing a patient. And to do this, we often do it in healthy human volunteers, and they're often given particular doses, defined milligrams of the drug via the oral route, and then subjects subjected to blood sampling. So blood samples are taken, say, once hourly or 2 hourly and out to 4, 6, 8, 12, 24, 36 hours. And those blood samples are then prepared for chemical analysis in a laboratory where technicians begin measuring drug concentrations in the blood sample using some sort of analytical methodology. Okay, we've come a long way in our techniques and the methodologies for doing this. So things like mass spectrometry are now standard, very powerful technologies for monitoring and analysing drug concentrations in tissues. And hopefully you'll get back from those technicians some data in the form of concentrations of drugs. So often it's expressed as micrograms. So remember, a microgram is 1,000th of a milligram, and a milligram is 1,000th of a gram, and a gram's 1,000th of a kilogram. So with tiny concentrations in micrograms per litre of blood. And essentially when we get that information, we plot all that information, and usually we're using a dozen or so patients. So we've got groups of data all at the same time point with the same dose, and we can average those values and plot them on a graph, and we get something like this, where we get drug concentrations in blood on the y-axis plotted versus time on the x-axis, where the x-axis is defined as time 0 equals the time of ingesting the drug. So we see a rise in blood concentrations as the drug undergoes that process of absorption and distribution, and it begins falling with time. Okay, so this is known as a blood concentration versus time curve or time profile, and they are absolutely gold. They are really extremely valuable in pharmacokinetics. Unless we have this information, and unless it's of a high quality scientifically, very robust. Really, we can't say anything much at all about the pharmacokinetics of drug, because unless we have this information relating blood concentrations versus time and dose, we really don't know how often to give a drug to a patient or how much of a drug to give to a patient. So we really do need this sort of very valuable information. So it's important to realise then that those 4 ADME processes that shape the pharmacokinetic properties of drugs also shape the morphology, the appearance of a blood concentration versus time curve. So here we have a drug being ingested by the patient. We see this rise in blood concentrations under the influence of absorption, and then as it reaches its peak concentration, we're seeing optimum or peak distribution of that drug into tissues and equilibrating between blood and tissues, and then we start to see those concentrations fall under the influence of those 2 processes that reduce drug concentrations in the blood: metabolism and excretion. Okay, so you can see now how these 4 fundamental processes have a lot to do in determining how blood concentrations rise and fall after dosing, and they've got a lot to do with whether you need to take a drug 3 times a day, twice daily, or once weekly. Now, also another thing that we're trying to determine when studying drug concentrations and time in patients is we're also trying to define a thing called the therapeutic window. And this is a really important concept, and some say it's really one of 2 fundamental concepts in pharmacology. You'll have to keep listening for another couple of videos to learn the other one. But the therapeutic window is really important. Therapeutic, of course, is a term that refers to healing outcomes or relief of disease symptoms. So we administer a drug to a patient and we see that process of absorption. The drug enters the blood. We see this rise in blood concentrations until they reach a certain maximum. Well, the idea is that the patient only begins experiencing benefit once the blood concentration reaches a certain minimum effective concentration. Okay, as the name suggests, if the concentration is lower than the minimum effective concentration, the patient won't produce any benefit. Okay, so there's no benefit in the patient while the blood concentrations are rising here until they reach this minimum effective concentration. And this is the bottom boundary of the therapeutic window. Once the drug level crosses that threshold, the MEC, then obviously the patient begins experiencing benefit. And but we also need to guard against giving too high a dose of the drug, because they might exceed the upper boundary of that therapeutic window, the minimum toxic concentration. So in this case, the drug might cause headaches or nausea and vomiting, 2 of the most common drug-induced side effects, if we exceed certain concentrations of the pharmaceutical in the blood. So we need to get the dose right so that the levels stay within this sort of sweet spot defined by the upper and lower boundaries of the therapeutic window. Okay, so essentially, patients only benefit from drugs when concentrations are within this therapeutic window. So let's imagine that you're working in your garden shed and you drop a hammer or an axe on your big toe. It hurts like mad. You rush inside and take some ibuprofen for the pain, and it takes 20 to 30 minutes before the pain starts to subside after taking the ibuprofen. And this is really just reflecting the time taken for absorption, the time for blood concentrations of the ibuprofen to reach this minimum effective level after the drug is released from the pill as it undergoes dissolution and disintegration and release its payload within the gastrointestinal tract. That then gets into the blood and then gets circulated around to the body, to the brain or to the injured tissue. Okay, so the patient begins benefiting once the blood concentrations exceed that minimum effective level. Then they'll reach some sort of peak effect when distribution is at equilibrium, distribution into tissues is at equilibrium, and then the blood concentrations start falling under the influence of metabolism and excretion, and then at some point the pain returns and your toe starts throbbing again because the blood concentration has fallen below that bottom boundary of the therapeutic window. So it's time to take another dose. Also of interest is the time taken to reach the peak. So the peak of the so-called Cmax, the maximum concentration in the blood, is really the point at which the patient derives maximum benefit from a given dose of a drug. Sometimes that's important. If you're waiting for relief of acute pain caused by dropping a hammer on your foot, obviously you want a drug with a short Tmax because you want to get that pain relief as quickly as possible. Or if you're suffering from a migraine headache, intense headaches, then choosing one drug over another because it has a shorter Tmax is sometimes appropriate. On the other hand, for a lot of conditions, if a patient's taking a blood pressure lowering drug, for example, they're going to live on that drug for a decade or more, then the Tmax isn't as important a factor in choosing one medication over another. So it depends a lot on the context of the clinical condition and the reasons why we're administering a drug. It's important to realise too that when we're doing these human studies of new pharmaceuticals, we're trying to work out what doses of the drug should be given to a typical human being to try and achieve concentrations within this therapeutic window. In this case, I've got a single pill being administered, and this is too low a dose because the maximum plasma concentration at this low dose doesn't achieve that minimum effective concentration to alleviate patient symptoms. So giving 2 pills in this case is about right, and but giving 4 is too high because the patient experiences side effects for some duration of the time after treating with the drug because it exceeds the upper boundary of the therapeutic window. Okay, so the bottom line of this is it really is important when you take a pharmaceutical to follow the directions on the packet or the product insert or the advice given by your pharmacist, because it's very important that the doses you ingest actually achieve concentrations within the therapeutic window, that you're not giving too low a dose because then you get no benefit, and even more importantly, you're not getting any harm because you gave yourself too high a dose of the drug. So whether it's a common everyday medication such as ibuprofen or acetaminophen, paracetamol, aspirin, all those sorts of medications, make sure that you use them at the recommended doses. They're there for a reason. We're trying to protect you, but also give you maximum benefit from your pharmaceutical. Things get a little more complicated when we're dealing with situations where you need to take repeated doses of the drug. And this also is a time when we need to get those doses right, because you can imagine with repeated dosing, depending on how quickly that drug is removed from the bloodstream by those processes of metabolism and excretion, it may actually take some time before the blood concentrations achieved by a particular dose reach into that therapeutic window. So just in this hypothetical example I've got here, the first couple of pills taken at time 0, the maximum concentration is below that therapeutic window, the lower boundary of the therapeutic window, the MEC. So the patient doesn't get any benefit at all from that first dose. And it's only when they take the second dose, and then a couple of hours later, perhaps the plasma or blood concentrations reach effective levels within the therapeutic window. And then hopefully the dose that we're giving to the patient will ensure that with repeated dosing, when we reach what's called steady state, where the amount of drug leaving the body and the amount of drug entering the body is essentially equal, is achieved with repeated dosing. Under those conditions, with long-term use of that drug, hopefully these peaks and also the troughs, as we call them, will both be within the therapeutic window, so the patient is benefiting from the drug all the time. Another important concept relating to the therapeutic window is how wide it is. A drug with a wide therapeutic window, where the minimum effective concentration is very well resolved and very different to the upper boundary, the minimum toxic concentration, is going to be safer for a patient than a drug with a narrow therapeutic window, where the minimum effective concentration and the minimum toxic concentration aren't too different. So a good example would be something like digoxin, a cardiac stimulant drug that's been used for centuries. And of course, it's used in treating people with what used to be called dropsy or congestive heart failure. But it's an extremely toxic drug. So while it's quite effective, it can be very toxic. So there's a very narrow margin of error associated with using digoxin. So it's a classic example of a drug with a narrow therapeutic window. And also, as you'd imagine, it's important that as pharmacology matures and as the pharmaceutical innovation sector grows in its scientific capabilities, that as we release new drugs onto the market within a therapeutic class, that is within a group of structurally related molecules that are used for the same purpose, for treating the same disease, that is, hopefully, even though the first so-called Gen 1 molecules, the innovator drug, the first drug in a class, first drug came available for treating a particular clinical condition, for example, they sometimes had an element of toxicity associated with them, even at regular doses. This is because they might have a narrow therapeutic window. And hopefully by the time the chemists in other drug companies or even the same pharmaceutical company come up with a second generation molecule, it'll have some improvements over the first Gen 1 drug, the innovator drug, so it has a wider or more intermediate therapeutic window, and hopefully by the time we get to a Gen 3 drug, that therapeutic window is much wider indeed, meaning that it can be used much more safely in patients. So that's sort of the hypothetical goal of the pharmaceutical innovation sector, and that's one reason why new medicines come on the market, hopefully, reasonably regularly, even when they're pre-existing members of that same class. Hopefully it's because later products within that class of medicines are safer for patients with a wider therapeutic window. Hopefully it's not just driven by the desire to make money, and there really is some clinical rationale underlying the use of introduction of a new medicine. Okay, we've covered a fair bit of ground, but let's just quickly recap before we wrap up this particular video. Pharmacokinetics is a branch of science. We named it, stealing that term from the chemists, simply because we needed a term to describe study of some very important properties of drugs, that is, what happens to them while they're in the body. And we've seen that has a lot to do with 4 fundamental processes: the readiness with which they're absorbed, distributed, metabolized, and excreted. And those fundamental processes have a lot to do with determining whether the medication needs to be taken once daily, twice daily, 4 times daily, or once monthly, or once weekly. These fundamental 4 processes have a lot of impact on the use of drugs in medicines in patients. Also, we've learned about the all-important blood concentration time curves. It's really important. Just before we can say anything sensible and intelligible about any drug, we need to have this sort of data, and it needs to be rigorous and well-defined and well-validated experimentally. And it's also important that we understand the effect of different doses of drugs on blood concentrations with time. Also, we've introduced this all-important concept in pharmacology of a therapeutic window, and that's simply a range of blood concentrations that we're trying to achieve when we administer a particular dose of a drug to a patient. We're trying to produce as much benefit to the patient, but also minimising harm. So that's the whole point of pharmacology. Unlike some scientific disciplines and fields of medicine that are very concerned with public health, that's all very noble. But in pharmacology, we want to protect individuals from the individual effects of drugs in terms of adverse effects. So it's very important that we administer them at doses that achieve blood concentrations that remain within the therapeutic window. And hopefully, we've also seen that the goal of ongoing pharmaceutical innovation is not just to keep the sector viable financially, which is very important, but it's also because we're trying to come up with new medications that achieve benefits for patients with lower risks. Okay, well, thank you very much for your attention. I really appreciate your presence with me, and thanks for watching this video. I will follow this one up. Please hit the subscribe button and the notification button in YouTube, and next time, God willing, we'll explore another important topic, one we've touched on today. Absorption is for drug, A is for drug absorption. That is the first of these physiological processes that determine the ADME properties of pharmaceuticals. Really important. Sounds pretty boring, but believe me, it's a fascinating topic, and we'll unpack some of its implications for the use of medicines in that episode. All the best, take care, and thanks again for watching. Bye-bye.